Okay, here is our big picture again of cellular respiration, and we've been looking at, let me get a pen here. So we've looked at glucose going into glycolysis and forming two pyruvate molecules, and that happened out in the cytoplasm. So those two pyruvate molecules then go into the mitochondria and into the bridge step, and coenzyme A attaches to 2-carbon molecule to form acetyl-CoA, and in that process carbon dioxide is lost and will just leave the cell as waste. And in the process, we got one of these high energy electron carriers. So let's see what those high energy electron carriers are for. So pyruvic acid, pyruvate, we lost 1 carbon to carbon dioxide, and then in the citric acid cycle, we lost the other 2 carbons as 2 carbon dioxide molecules. So our glucose molecule is totally broken down. And so that is cellular respiration is a catabolic process. And the way I remember that is this is compared to an anabolic process. So a catabolic process is one where a molecule is broken down to get energy. An anabolic is where a molecule is built up to store energy. I always think of catabolic as being, I remember it as breaking up the molecule because it's like sort of a catastrophe or an explosion, a catastrophic explosion. So I remember catastrophic as breaking up. The glucose molecule is a catabolic process. And that's what happened. Our glucose molecule has been broken up and all the carbons are gone, and what we've got are these high energy electron carriers, and we've got just a little bit of ATP. Okay, so what happens now? Where do those high energy electron carriers go? They go into the electron transport chain. Okay, and this is really cool. So what's going on here? This picture, there's a lot to it. First thing you'll notice is these are proteins in a membrane. So they are in the inner membrane of the mitochondria in eukaryotes. This also happens in the cytoplasmic membrane of bacteria or eukarya. So it doesn't have to be in an organelle. This happens in both eukaryotes and prokaryotes. Okay, so here's our membrane. You hopefully recognize these molecules right here. These are our phospholipid molecules that are making up our phospholipid bilayer. And these are proteins, enzymes that are catabolizing the reaction. Excuse me, did I say that right? Catalyzing the reaction. Catalyzing the reaction. Okay, and here's our high energy electron carrier. They are carrying the stars of the show. Our stars are in here. And so this first enzyme, this first protein right here, will take those stars, those high energy electrons will be transferred into this protein. And in the process, NAD+ will come off, and also that hydrogen ion. So it's now a hydrogen ion, that's now floating around out here in the cytosol, or cytosol. I think it's still called the cytosol. Inside the, it's make sure it's inside the mitochondrion space in here. Okay, so what are these, the stars of our show? What are they doing? Well, when these high energy electrons, and here they are right here, our high energy electrons are transferred into this protein, they attract these hydrogen ions because the electrons are negatively charged. So they attract these positive ions. And so the positive ions will move in, attracted by those electrons. At the last minute, they're fooled because this protein right here, called it ubiquinone, it's a mobile carrier, will come over and take those electrons away. And there's nowhere else for those proteins to go, but they get transported to the other side of the membrane. So this whole system right here is for transporting hydrogen ions across this membrane. And it does it again. This mobile carrier will take these electrons to the next protein. And the same thing will happen again. These hydrogen ions will be attracted to the negative charge, and boom, this other protein comes along and steals the electrons away, and the hydrogen ions get transported across. And here it happens again. So the electrons get transported to the next protein, and here they are. Here's our hydrogen ions being attracted to those negative charges, and boom, those electrons go to the next protein, and the hydrogen ions get transported across. So why? Why all this trouble just to be able to transport hydrogen ions across this membrane? Well, what this does is it sets up a concentration gradient. So let's write that down. So this is a concentration gradient. There is an I. There's my I. Gradient. And this is potential energy. So now we have hydrogen ions over on this side, and lots of them. So this is high hydrogen ion concentration on this side, low hydrogen ion concentration on this side. So they would like to go across this membrane and go into lower concentration. But there's this membrane that's in between them, and the ions, because it's a full charge, those ions cannot get through that semi-permeable membrane. It is not permeable to hydrogen ions. So the only way that they can get through is there's a protein right here that will allow them to go through. So they will go through this protein by diffusion, or this would be facilitated diffusion because it's using a protein. So facilitated diffusion. And I don't have enough room here. I'm abbreviating that. That's facilitated diffusion. So what about this? These hydrogen ions were being transported across using the energy from the electrons. So that would be active transport. But in the process of these hydrogen ions flowing through this protein, it ends up turning this wheel. The protein, it's like a wheel. and it acts almost like a water wheel on a river. So, and just imagine a big water wheel on a river, and imagine it turning around and around and around and around, and you could get energy from that to grind grain historically. But in this case right here, the energy of turning the flow of hydrogen ions through here, and from an area of higher concentration to an area of lower concentration, turns this water wheel, and the energy is being used to make, guess what? ATP. Yeah, and a lot of it. So, let's see. It's how much ATP? So it's 34. I believe. Let me check my notes just to make sure. Let me get that right. Yeah, 34. Okay, so 34. Did I leave? Here I am back again. Okay, so 34 ATP. But there's a problem. These electrons are kind of stuck in here. Where do they go? Where are they supposed to go? Well, there needs to be something to pick them up here at this end. If there isn't anything here to pick up those electrons, it becomes like a bucket brigade that gets stopped. Let's say you've got a bucket brigade of people trying to put out a fire, and you've got a well. At one point you've got the fire. Let's see, you've got the fire at the other point, and then you've got a bucket brigade of people in between handing off the buckets until you finally put out the fire. Well, it's like the last person just stops and doesn't empty their bucket. So it stopped. And that would be like this. That means it wouldn't continue. The last person, the person near the well, wouldn't be able to pick up another bucket of water until they can get rid of their water. So oxygen comes to the rescue. Oxygen is the final electron acceptor, and that's important. Got to know that. Final electron acceptor. So it accepts those electrons so that another NADH or another FADH2 can drop off their electrons and go through this whole process to make this concentration gradient and allow the hydrogens to go through. So another player in this that's really important to know is this enzyme right here, ATP synthase. And that seems pretty easy to remember, ATP, and you're synthesizing ATP, so ATP synthase. Okay, so oxygen is our final electron acceptor. So oxygen accepts those electrons and accepts 2 hydrogens in the process to form water. And there is our last product that we needed to account for. This is the water, the waste that's coming off. So water, so it's O2 plus our hydrogens gives us water. I believe 2 water. It's 2 waters. Okay. Oh, I see, because they've got a half right there. That's fine. Okay. So I hope you found that interesting, and that's cellular respiration.